Welcome to Developmental Biology. Today, we are exploring one of the most revolutionary breakthroughs in modern reproductive medicine, in vitro fertilization, commonly known as IVF. We will understand both the biological miracles and the mathematical realities that make this life-changing technology possible. Whether you are completely new to this topic or looking to deepen your understanding, we will build everything from the ground up. Let us dive in. To start, let us imagine you are trying to grow a delicate plant from a seed, but the soil in your garden is currently too harsh. Instead of leaving it to chance, you might plant it in a perfectly controlled greenhouse pot, give it exact nutrients, and wait until it sprouts into a strong seedling before transplanting it back. In vitro fertilization works exactly like this greenhouse analogy for human reproduction. The term in vitro literally translates to in glass, referring to the laboratory dishes where this delicate fertilization process takes place. When natural conception is difficult, doctors create a perfectly controlled environment outside the body to help the egg and sperm unite. Now that we understand the basic idea, let us look at how the actual medical procedure unfolds. IVF is not a single event, but a complex series of steps. It begins with ovarian stimulation, where specific hormones are administered to encourage the ovaries to produce multiple mature eggs, rather than the usual single egg. These eggs are then surgically retrieved and carefully mixed with sperm in a specialized laboratory setting. Once fertilized, the eggs develop into embryos over a few days under strict, round-the-clock observation. Finally, one or more of the healthiest embryos are transferred directly into the patient's uterus. If the embryo successfully implants into the uterine lining, a pregnancy begins, continuing exactly as it would in a natural conception. Before we move on, let us see this entire process visually. This diagram maps out the complete IVF journey. Starting at the top, you can see ovarian stimulation, where hormones act on the follicles. Following the blue arrow to the right, we see the egg retrieval process using a specialized needle. Down at the bottom is the fertilization stage, taking place in a laboratory dish. Moving to the left, the fertilized egg enters the embryo culture phase inside a temperature-controlled incubator, developing from a single-cell zygote into a multi-celled blastocyst. Finally, the cycle completes in the center with the embryo transfer, where a catheter carefully places the blastocyst into the uterus, specifically targeting the endometrium. Having seen how the procedure works, we must ask, why is this technology so crucial? IVF is widely used to overcome various biological obstacles that make natural conception impossible, such as blocked fallopian tubes or severe male factor infertility. But its applications go far beyond just treating infertility. IVF is absolutely crucial for genetic screening, allowing doctors to test embryos for severe hereditary diseases before implantation, ensuring a healthier start for the child. It also plays a vital role in fertility preservation. For example, it enables cancer patients to freeze their eggs or embryos before undergoing harsh treatments like chemotherapy that might otherwise leave them sterile. While IVF is fundamentally a biological process, understanding its clinical success requires a bit of mathematical probability. It is important to realize that a single cycle of IVF does not guarantee a pregnancy. There are countless biological variables that can affect the outcome at multiple stages, from egg retrieval to implantation. Because of this reality, doctors use a cumulative probability formula to help patients understand their true chances of success over multiple IVF attempts. This mathematical model is not just for scientists. It is essential for counseling patients, helping them manage their emotional expectations, and planning the financial aspects of their treatment journey. This brings us to the mathematical model used by fertility specialists. The formula is P sub n equals 1 minus the quantity 1 minus p, raised to the power of n. Let us break down what each symbol means. P sub n represents the cumulative probability of achieving at least one successful pregnancy after a specific number of cycles. The variable p represents the probability of success for a single individual IVF cycle, which depends heavily on factors like the patient's age and overall health. Finally, the exponent n represents the total number of IVF cycles the patient undergoes. As the number of cycles n increases, the cumulative success rate, p sub n, also increases. Let us see this formula in action with a concrete example. Imagine a couple consulting with a specialist. They learn their chance of success per single cycle is exactly 30%, and they commit to 3 complete cycles. First, we identify our given values. P is 0.30 and N is 3. We cannot simply add 30% 3 times. Instead, we calculate the failure rate of a single cycle, which is 1 minus 0.30, giving us 0.70. Next, we raise this failure rate to the power of 3 to find the probability that all 3 cycles fail consecutively, which equals 0.343. Finally, we subtract this from 1. The result is 0.657. This means the couple has a 65.7% chance of achieving at least one successful pregnancy. Now that we have used this formula, let us understand where it comes from logically. Why do we subtract from 1, and why do we use exponents? We start by defining P as the probability of success in a single cycle. By the basic rules of probability, if success is P, then the probability of failure in that same cycle must be exactly 1 minus P. Now, each IVF cycle is an independent event. The outcome of the first cycle does not change the biological odds of the second. To find the probability of multiple independent events happening in a row, we multiply them. So the probability of failing n times consecutively is 1 minus p multiplied by itself n times. Continuing our derivation, multiplying 1 minus p by itself n times gives us the expression 1 minus p raised to the power of n. This represents our absolute worst-case scenario, total failure across all attempts. Now, in probability, the sum of all possible outcomes must equal exactly 1, or 100%. The only alternative to failing every single time is succeeding at least once. Therefore, to find the probability of at least one success, which we call P sub n, we simply take the total probability of 1 and subtract the probability of total failure. This leaves us with our final formula. P sub n equals 1 minus the quantity 1 minus p to the power of n. We have looked at the overall success of a cycle, but what happens inside the laboratory during that cycle? The IVF process experiences natural biological attrition. Not every mature egg retrieved from the patient will successfully fertilize when mixed with sperm. Furthermore, of those that do fertilize, not every single one will develop into a viable blastocyst suitable for transfer. Because of these successive drop-off rates, doctors can't just retrieve one egg if they want one embryo. They must work backward from their final target number of embryos using historical conversion rates to estimate exactly how many initial mature eggs they need to retrieve. To calculate this starting point, clinics use a straightforward algebraic equation. The formula is B equals E times RF times RB. Let us define these variables. B represents the final target number of viable blastocysts the doctor wants to guarantee. E is the unknown initial number of mature eggs that we need to find. The symbol RF stands for the fertilization rate, which is the percentage of eggs that successfully combine with sperm. Finally, RB represents the blastocyst development rate, which is the percentage of those fertilized eggs that survive to the final embryo stage. Both rates are used as decimals in the calculation. Let us apply this equation to a real clinical scenario. A doctor wants to guarantee a yield of exactly 4 viable blastocysts. Historical data shows an 80% fertilization rate and a 50% blastocyst development rate. We start by identifying our knowns. B is 4, RF is 0.80, and RB is 0.50. We plug these into our equation. 4 equals E times 0.80 times 0.50. Multiplying the two biological rates together gives us a combined overall efficiency of 0.40. Finally, we divide our target of 4 by 0.40 to isolate E. The result is 10. The doctor must aim to retrieve exactly 10 mature eggs to expect 4 viable embryos. We have covered a lot of ground today, so let us review the most important ideas. First, in vitro fertilization is a profound application of developmental biology that bypasses natural reproductive barriers by facilitating fertilization in a highly controlled laboratory environment. Second, we learned that the process is a carefully managed sequence of ovarian stimulation, egg retrieval, fertilization, embryo culture, and transfer. Third, because biological variables exist, we use cumulative probability models to realistically predict success over multiple cycles. Finally, understanding biological attrition allows doctors to calculate exactly how many eggs to retrieve. You now understand both the biological mechanisms and the statistical realities of this life-changing technology.